Welcome everyone to the 5th webinar in our 6-part series on alternative approaches for acute inhalation toxicity testing. My name is Amy Klippinger. I'm the Associate Director of the PETA International Science Consortium and will be co-moderating today's webinar with the USNTP Interagency Center for the Evaluation of Alternative Toxicological Methods, also known as NICETM. I'd like to thank you all for joining this webinar, and before I introduce today's speakers, I'll quickly run through a few logistics. The slides and a recording of today's webinar will be posted online at the link on your screen within a few days, and the previous 4 webinar recordings are also posted there as well. Everyone is currently muted. If you have a technical question for me anytime during the presentations, you can use the question box, which is located in the GoToMeeting panel on the right-hand side of your screen. If you have a question for one of the speakers, you can type it in that same question box, and we will ask your questions to the speakers during the Q&A period. So please get them in anytime during the webinar, and we'll ask them all after both of the presentations are finished. I want to point out that this webinar series is being co-organized with input from multiple organizations, which are shown on your screen now, and I wanted to just take a moment to thank each of them for their efforts. And very quickly, for those of you who are interested in developing adverse outcome pathways, I wanted to mention an opportunity being co-organized by my organization and the EPA to encourage new users to add information to the AOP wiki. And I won't go through the details of this award, but you can visit the link shown here on the screen for additional information. And lastly, before introducing today's speakers, our last webinar in the series will be held on September 8th. On September 8th, Drs. Dan He and Kelly Berube will be speaking about 21st century approaches to inhalation toxicity testing, more specifically their lung on a chip and 3-dimensional in vitro lung model work. And I hope you'll all be able to join that webinar as well. So with that, our first speaker today is Dr. Matthew Vinken. Matthew is a professor affiliated with the Free University of Brussels in Belgium and the University of São Paulo in Brazil. He has a background in pharmaceutical sciences, holds a doctoral degree in vitro toxicology, is a European registered toxicologist, and a trained chemical risk assessor. He is vice president of the European Society of Toxicology in Vitro and associate editor of the journal Toxicology in Vitro. His expertise lies in liver-based in vitro modeling, liver toxicity, and mechanistic toxicology. Matthew?
Yes, thank you very much for this nice introduction, Amy, and I also would like to thank you for inviting me to give this presentation today. So I hope you all see my screen. So I rely a little bit on Amy for technical emergencies. So if there would be a technical emergency, please contact Amy, and I will ultimately also get the message, I assume. So I would like to give you an introduction, a brief introduction into the area of these adverse outcome pathways, these AOPs, and I would like to discuss how they can be used as tools in the fields of toxicology and risk assessment. Now, before doing so, I would like to sketch a little bit of the context or the background. So I would like to discuss quite briefly how exactly they can introduce into these areas. Well, I think that especially the toxicologists among the audience know very well that there has been a tendency in the last few years to turn more to mechanistic toxicology. So rather than the classical toxicology in which the toxicologists have for years produced apical toxicological endpoints without really deeply wondering about the mechanistic basis that was driving these apical toxicological endpoints. Now, a first relevant concept that has been introduced in this context is the so-called mode of action, which is defined as a series of key events along a biological pathway from the initial chemical interaction to the adverse outcome. Now, the mode of action was first used in the cancer field, but over time it has also been applied to non-cancer endpoints as well, such as, for example, has been the case for ILSI, so the International Life Sciences Institute, but even at the level of the World Health Organization. So basically, the mode of action has been mainly, if not uniquely used for the field of human health risk assessment. And this is in contrast to the AOP, the adverse outcome pathway, that has its roots in the field of ecotoxicology. And only in the last few years, it has also begun to be introduced into the field of human health risk assessment. Now, a first milestone, if you will, in this regard was the publication of, well, the document that we all know very well that has been published by the US National Academy of Sciences, so outlining a vision on toxicity testing in the 21st century, in which so-called toxicity pathways played a very important role. And actually, a toxicity pathway is the basis, if you will, for an AOP, which is defined as a conceptual construct that portrays existing knowledge concerning the linkage between a direct molecular initiating event and an adverse outcome at a biological level that is actually relevant to the field of risk assessment. The second document that has been published here, and that is of relevance, has been issued by the OECD and is a kind of a guidance, if you will, on how to develop new AOPs and especially how to evaluate newly postulated AOPs. So you can find the reference here below on this slide. It has been turned, I think, 2 years ago into an AOP handbook, which can be found on the AOP wiki. And actually, this will serve as the basis for the remainder of this presentation. So on this slide, you can see the link between a toxicity pathway, a mode of action, an AOP, and a source to outcome pathway. And this is illustrated by means of an example taken from the field of ecotoxicology. And you can immediately see that an AOP indeed spans over different levels of biological organization, which is fully in line with the definition as discussed on the previous slide. And you can also see that it is more comprehensive in comparison with the mode of action, as it can start with the exposure and it may go all up to the population level. It does not necessarily has to, but it can go all up to that level. And in turn, a mode of action as shown here is a broader concept in comparison with the toxicity pathway. So this is how an AOP roughly looks like, and this is what I like to call the macrostructure, if you will. Of course, this is not official designation, that's my own interpretation. But anyhow, each AOP consists of two so-called anchors. So the first anchor is the starting point, which is called the molecular initiating event, the MIE, and that denotes the interaction of a chemical with the biological system. And then the second anchor is the actual endpoint of the adverse outcome pathway, the adverse outcome as such, which denotes the apicotoxicological endpoint. And the entire space between these two anchors, as you can see here on this slide, is filled with so-called key events, which are interlinked by key event relationships. Now, I will be coming back to this further on, but until about 2 years ago, the OECD also made a distinction here between key events and intermediate steps, which were another form of information blocks that also were included in the matrix between the MIE and the AO. Now, they have given up this distinction between a key event and intermediate step. I will be coming back to this further on because I think it can still be of value, but I already mentioned it now. So if we then have a closer look to an AOP, the microstructure, if you will, you then can see indeed, as shown on this slide, that again, in line with the AOP definition, all of these information blocks span over different levels of biological organization. So there can be effects at, for instance, a cellular level, like protein production, for example. An outcome at the organ level, like disrupted homeostasis. Another example is an effect at the organism level, like impaired development. And in the most extreme cases, there are also effects at the population level, like extinction of entire species. But of course, as you already can feel, this mainly applies to ecotoxicological AOPs. Now, in the next set of slides, I would like to briefly discuss on how to develop an AOP, and this is done in accordance with this OECD guidance. And actually, there are two strategies here. So you can actually start from the starting point, which is called a top-down procedure, or you can start from the second anchor, which is the apicotoxicological endpoint, and this is called a bottom-up approach. So the first step in the top-down procedure, or the second step in the bottom-up approach, if you will, is the identification of the AO, which is defined as a change in morphology, physiology, and so on of an organism or system that results in the impairment of the functional capacity, or at least the capacity to compensate for stress. So here again, as holds for all these information blocks in AOPs, they can be located at different levels of biological organization, as exemplified on this slide. And also very important to mention here is that an AO, so an adverse outcome, can refer to a systemic effect, like for example is the case in reproductive toxicity, but it can also refer to a local effect, like skin irritation, for instance. And then the second step in the top-down procedure, or the first step in the bottom-up approach, if you will, is the identification of the MIE, which is the initial point of chemical-biological interaction within the organism. Now, very, very important here is that you define the site of action of the MIE, because you can imagine that an adverse outcome, so the actual apicotoxicological endpoint, can be very, very different depending on the site of the MIE. And this specifically holds true for these receptor-ligand interactions, because as you probably know, we usually have receptors in different kinds of tissues, which then results in different effects. So that is an example of an MIE. Another one could be a protein oxidation or DNA binding. The next step in AOP development is the identification of the key events. In other words, a change in the biological state that is both measurable and essential to the progression of a defined biological perturbation leading to the AO. So basically, as discussed already before, this defines the entire matrix between the two anchors, the MIE and the AO. So now I will be coming back to what I already mentioned before, so this distinction that the OECD made until a couple of years ago between a key event and an intermediate step. So an intermediate step in this context was an information block, also included in an AOP, but that in contrast to a key event was not necessarily measurable and did not as such really was essential for the AOP and the AODAS to occur. Again, I will be coming back to this further on at the end of the presentation when I will be discussing the applications of AOPs specifically in the context of risk assessment. Now, as such, there is no restriction in the selection of the number of these key events, but of course they should make sense, which means that you should have a good understanding of the normal physiological and especially the pathological pathways that they underlie. Well, as you can imagine, there are several ways for the identification of these key events. The most obvious thing, of course, is going through all of the currently available and relevant scientific literature. And this also explains why it is usually quite some work to establish a scientifically sound AOP. But in fact, that's not the only thing. What you're supposed to do is to include and to consider any piece of relevant information in AOP development. So this could be animal tests, so in vivo data, in vitro data, in chemical data, and silico data, and especially in the last few years, all this type of different omics-based data. And then the final step in AOP development is description of the key event relationship, so that define a direct relationship between two key events and that facilitate the extrapolation of the state of a downstream key event from the upstream key event. Now these key event relationships may refer to a direct between a pair of key events that are adjacent in an AOP, or they can denote an indirect linkage between a pair of key events for which a relationship is thought to run through a number of another key event or a gap in current understanding. Now the reality is that in most, if not all of the currently available AOPs, that these key event relationships are only qualitative in nature. Now, of course, as you can imagine, if we want to apply AOPs for risk assessment, now that's exactly what we want to do, of course, we should strive for key event relationships that are quantitative in nature, and this can be accomplished in a number of ways. So, for example, by considering dose response relationships, also by including relevant thresholds or points of departure, mathematical equations can be of use, or also biologically based computational models. And finally, also to mention here is that these key event relationships may be influenced by a number of modulating factors, which is specifically the case for ecotoxicological AOPs. So as you can imagine, this AOP development is quite a subjective exercise. So basically, the author of an AOP decides what piece of information can actually be included in an AOP. Now, to somehow check for the reliability and the robustness of a newly postulated AOP, the OECD has proposed two strategies. So the first one is a weight of evidence assessment, which should be done by meeting the so-called Bradford-Hill criteria. Now, without going into the details, this Bradford-Hill criteria, this is something that comes from the field of epidemiology and that define the basic criteria that should be met in order to establish a causal link between events, so in this case, between different information blocks of an AOP. And then you end up with a bunch of questions that you need to answer, like what about the concordance of those response relationships? Did you check the temporal concordance among the key events and the AO? Is there something known about the strength, the consistency, and the specificity of the MIE-AO association? What about biological plausibility, coherence, and consistency of the experimental evidence? Are there any alternative mechanisms that might interfere with the AOP? And finally, are there any uncertainties, inconsistencies, or data gaps left to be filled? And then the second kind of strategy that should be in parallel is a so-called confidence assessment, which should be done by answering a couple of so-called key questions that have been defined by the OECD themselves, namely how well characterized is the AOP, how well are the MIE and the key events causally linked to the AO, what are the limitations in the supporting evidence, is the AOP specific to certain tissues, life stages, or age classes, and finally, are the MIE and key events expected to be conserved across species. So each author of a newly proposed AOP is assumed to come up with a scientifically sound and a quite elaborate answer to each of these questions. And I can assure you from my own experience, if you want to do this in a correct way, that this is quite some work. So in the next few slides, I would like to show you some examples of already fully adopted and developed AOPs. And the first one has been developed by OECD and is about chemical-induced skin sensitization. Now, as some of you probably know, skin sensitization is a process that consists of two major events. So in the first event, which is called the induction phase, the first contact of the skin with the chemical takes place and a kind of an immunological memory, if you will, is being created. And then there is a second event, which is called the elicitation phase, in which the next contact of the skin with the chemical takes place and the actual clinical effects are known to be manifested. Now, a very important event here is the penetration of a chemical into the skin where it interacts with local proteins, which is called haptenization, and this is shown here on the left-hand side. And actually, a haptenized chemical can then be taken up into cells and it can induce a number of processes, one of them being activation of inflammatory cytokines in keratinocytes. And at the same time, there is also the activation of dendritic cells. Now, these activated dendritic cells then move to the lymph nodes where they present the chemical or the antigen, if you will, to T cells, resulting in the proliferation, so the cell growth of these T cells. So what happens upon subsequent exposure of the skin to this chemical, so the elicitation phase, if you will, well, then this fully grown T cells will directly move to the skin where they immediately will interact with the haptenized chemical, and this will trigger all of the typical clinical effects that we know to be associated with skin sensitization, like, for example, the redness of the skin and the skin rash. So that was the first AOP. The second one is about liver toxicity and has been introduced already a couple of years ago by the JRC, so the Joint Research Center of the European Commission, but only very recently it has been submitted for publication to Toxicological Sciences. It is shown here. So in this case, the MIE, the starting point, which is shown on the right-hand side, is protein alkylation and covalent protein binding, which then leads to hepatocyte injury, mainly by apoptosis, linked with inflammation and oxidative stress. The next step is then activation of Kupffer cells, which then triggers a very important, I would say the most important key event here in this AOP, namely the expression of transforming growth factor beta-1, which then drives the activation of stellate cells. And then the next step at the tissue level is the accumulation of collagen and changes in extracellular matrix homeostasis in the liver. So in other words, liver fibrosis. And I also want to draw your attention here to the different ways in which the different information blocks of the AOP are interlinked with each other. So then we are talking about these key event relationships, so the different arrows that you can see here. So the third example is another example of a kind of liver toxicity, again, which has been proposed by the JRC. But in this case, it's focused on liver steatosis, and in this case, the MIE, so the starting point is the activation of a specific nuclear receptor called the LXR, the liver X receptor. And this induces a broad set of transcriptional changes, all which are related, of course, to lipid homeostasis. Well, this not only then leads to de novo fatty acid synthesis, but at the same time also to the increase of fatty acid influx from peripheral tissues, which altogether then result in the accumulation of triglycerides in the hepatocytes, which are the most important cells in the liver. And then the next step at the organelle level is cytoplasm displacement, nuclear distortion, and mitochondrial disruption, which then leads to the manifestation of these fatty liver cells, so in other words, steatosis. Of course, these were only 3 examples that have been selected here because either I was involved in their development or I was also very interested because I have like an interest in liver toxicity as such. But of course, it should be clear that there are many more AOPs, and then I refer to the AOP Wiki, which has already been mentioned. So the AOP Wiki is a kind of a joint initiative in which the OECD has played a very important role, but of course it also involves a number of other agencies like EPA and so on. So what you see here on this slide is just a couple of examples of AOPs that have been included in the AOP Wiki. I just selected them at random. I think there are about 50 to 60 AOPs already in the AOP Wiki, but it's only to illustrate to you that it really concerns a broad variety of apical-toxicological endpoints and that it is not limited to human toxicology, but also to ecotoxicology, like for example the last one, fish cardiotoxicity induced by TCDD. Now, in preparation of this webinar, I have been looking in the AOP Wiki for AOPs relating to acute inhalation, but at least in my hands, I couldn't find any. So it might be a good time, actually, to come up with this kind of AOP. Now, very frequently, when I give this kind of presentation, I usually get 2 remarks. So the first one is, well, this is all very nice, but it's not new. And the second one is, well, again, it's interesting, but what can you actually now do with this? Well, as an answer to this first remark, so indeed, AOPs are conceptually not new because we already had like the mode of action, although there is somewhat a difference. But this is the first time that all of this is actually done in a kind of a context and a template that is globally accepted. So that has also been supervised now by the OECD. And then regarding the second remark, well, I hope that with this and the following slides, I will be able to convince you that AOPs can be of real relevance and can serve a number of purposes of relevance of the field of toxicology and risk assessment. The first one being chemical categorization and chemical grouping, which mainly focus on the MIE, so the starting points of the AOPs. Well, I think that especially the toxicologists among us know very well that specific chemical groups have been associated with the induction of specific toxicological endpoints. So this can serve as the basis for read-across approaches, which in turn can be fed into the structure-activity relationships, and if you're very lucky, quantitative structure-activity relationships. And actually, first example here comes from the AOP on skin sensitization, which, as discussed before, relies on a nucleophilic electrophilic reaction, so that's the MIE, that is typically triggered by aldehydes, epoxides, amides, and lactams. Second example is the AOP on steatosis, whereby the MIE was the binding of this nuclear receptor, the LXR, and this is usually seen with chemicals that have phenyl rings, chlorogroups, and methyl moieties. So that was the first example of an application of an AOP. The second one is test development, whereby the focus is not only put on the MIEs, but actually also on the key events, so basically on all of the information blocks in an AOP. So actually, these information blocks in an AOP, they define a number of biomarkers, most of which are in vivo relevant, which can be used as a kind of an inspiration, if you will, for the development of in vitro methods and by extension also in silico methods that can be used for the testing of a specific AOP. And here again, the example comes from the chemical-induced skin sensitization AOP, whereby, as explained a couple of slides ago, one of the most important key events is the activation of these dendritic cells. Well, actually, based on this knowledge, at least 2 tests, in vitro tests, have been introduced and developed in the last few years that really are looking for this dendritic cell activation as being a key event in the AOP. The first one is the myeloid U937 skin sensitization test, maybe better known under its acronym MUST. So in this kind of test, a specific human cell type is exposed to the chemical of interest, so of which you expect, or at least you suspect, that it might induce skin sensitization for a given period of time, after which the expression of CD86 is being investigated by means of flow cytometry, CD86 being a co-stimulatory receptor that is indicative for dendritic cell activation. And then there is a second, a very similar test, the HCLAT test, so the human cell line activation test. So here again, another cell type exposed to the chemical of interest, but in this case not only CD86 but also CD54, another indicator of dendritic cell activation, is investigated. So this is a very nice example of 2 tests that have been based on an AOP and specifically on the identification of a key event in an AOP. Now, AOPs also sometimes are called the embodiment, if you will, of integrating testing strategies. So they can be used for this purpose, as proposed also in the European chemicals legislation, REACH, of which some of you are probably familiar with. And of course, this contributes to the further implementation of the 3 R's concept into the safety testing of chemicals. I will be coming back to this further on. Now, AOPs can also serve a number of other purposes, one of them being the prioritization of chemicals, in this case specifically the identification of positive controls for a number of selected in vitro toxicity tests. And 2 examples taken from scientific literature in which AOPs have been used for the identification of benchmark chemicals of this kind of assays, or paraoxyon, which has been identified as being a benchmark chemical for in vitro developmental toxicity testing, and then the molic acid, which can be used as a positive control for a very specific in vitro neurotoxicity test. And then finally, AOPs, of course, can be used for several other purposes, very specific ones like the OECD test guideline program, which has a kind of regulatory relevance. Now, I have included this slide just for your information, but to make you aware that a lot of things are currently ongoing in the AOP field around the world. So as mentioned already a couple of times, a key role is really played here by the OECD. So not only have they introduced this guidance on AOP development and evaluation, and also they are co-founders of this AOP wiki, but also they have drafted a number of pathway-targeted case studies. In the US, specifically in the Hamner Institute for Health Sciences, focus is put on these estrogen-related and peroxisome-proliferated signaling, also in the context of pathway-targeted case studies. Also in the US, in the Center for Alternatives to Animal Testing, where specific attention is being paid to so-called pathways of toxicity, POTs, and then the entire collection of a POT, which is called the human toxome. And it is specifically looked whether omics-based information can be used for POT development. And then in Europe, we had this huge project, a collaborative project called SURAT, and SURAT is an acronym standing for Safety Evaluation Ultimately Replacing Animal Testing. And here we specifically addressed liver toxicity, and we looked, or we paid attention to possible applications of AOPs for the establishment of in vitro test methods, and also for the identification of biomarkers that can be used for in vitro toxicity screening purposes. I would like to elaborate a little bit on this latter project, so on SURAT, and I would like to use this to illustrate also what the next step is. So we first had AOP development, then AOP evaluation or assessment, if you will, and the next step now is AOP optimization. So before doing so, I would like to give you some background also about this SURAT project. So SURAT was raised in response to the European Cosmetics Regulation. I think that everybody is familiar with this, which actually foresees the testing ban, so we can no longer use in Europe animals for the safety testing of cosmetic products and their ingredients. And in addition, there is also the marketing ban. So CERET was a public-private research initiative between the European Commission on the one hand and Cosmetics Europe on the other hand. So they both provided 25 million euros, which was quite some money, of course. So CERET started in 2011 and was finished a couple of months ago in December 2015. It involved more than 70 different research institutions, most of them being European ones, and it was organized as 6 projects and 1 coordinating action, which is maybe better illustrated on this slide. So the idea behind CERET was to come up with a kind of a strategy to gradually replace the use of animals for repeated dose toxicity testing of cosmetic ingredients. Now, at the start of the project, we defined a number of so-called building blocks that were important to establish this strategy, and each of these building blocks has been embedded into a specific project as illustrated here. Because of time restrictions, I will not go through all of the details. If this would be of interest to you, you can have the web address below. But just to mention here that most of the AOP work has been performed in the context of this third project, Detective Project, which was about the identification and investigation of human biomarkers of repeat dose toxicity. Now, at the start of the CERET project, we wanted to know which organs can be potentially affected by cosmetic ingredients and the kind of toxicities that are involved in this process. And the major resource that we have consulted in this respect are the safety evaluation reports that have been published by the Scientific Committee for Consumer Safety, the SSCS. Some of you might be familiar with this. So the SSCS is a committee that at the European level takes care of the safety evaluation of cosmetic ingredients might be then included in the annexes of the European Cosmetics Regulation. So the SSCS publishes all these safety evaluation reports, which are called opinions, on their website, so you can freely download them. It's publicly available. And what we have done is we went through all of these opinions that have been issued between 2000 and 2009, about 250 different ones that cover in total about 220 cosmetic ingredients. And of course, in view of the CERET objectives, we have paid most of our attention during this screening exercise to information related to repeated dose toxicity testing. Now, if this would be of interest to you, we have published the outcome of the screening exercise. You can find the reference below. It was published in Archives of Toxicology. But just in a nutshell, we found that the liver is potentially the most frequently targeted organ by cosmetic ingredients, followed by the kidney and the spleen. And that liver toxicity is hereby mainly manifested by steatosis, but especially by cholestasis. And based on this knowledge, we then decided to, first of all, develop an AOP on cholestasis, because at that time that was not available. And that was then the basis for another publication, of which you can find the reference here below. It was published in 2013 in Toxicological Sciences. And what you see here on this slide is the AOP that also has been submitted to the AOP wiki. So I will just briefly guide you through it. So as you can see here on the left-hand side, the most important MIE is shown in a dark blue box, which is the inhibition of the BSAP, standing for the bile salt export pump. That's a specific drug transporter. And this leads to the accumulation of bile acids, which is shown in a dark green box. And actually, all of the dark green boxes in this AOP represent key events, while all of the light green boxes represent intermediate steps. So at that time, the OECD still had this distinction between key steps and intermediate steps, key events at least, and intermediate steps. So in the first instance, the accumulation of bile acids induces a direct deteriorative response, which is shown in yellow, and that goes hand in hand with quite generic processes like oxidative stress, inflammation, cell death, all which result at the organ level in the increased release of a number of enzymes into the serum like ALT, AST, but especially in the case of cholestasis, ALP, alkaline phosphatase. In a second instance, the accumulation of bile acids also drives an adaptive, a secondary response, if you will, which is shown here in purple, and that fully relies on the activation of at least 3 nuclear receptors. Again, this is a key event, namely the FXR, the PXR, and the CARB. Without going into the details, the activation of these 3 nuclear receptors induces a broad repertoire of transcriptional changes, all that are focused towards the increased removal of bile acids and their products from the system. So this is shown here in the middle of the AOP. Now this not only induces or actually increases the levels of bile acids in the urine, but also in the serum, and this in turn underlies the onset of the typical features that we know to be associated with cholestasis, like pruritus, so the itching, and the jaundice, so the yellowish pigmentation of the skin and also on the eyes. And here again, I also want to briefly draw your attention to the different key event relationships. So there's a kind of a color code here for the arrows, so referring to direct or indirect effects. If this would be of interest to you, I can only refer to the paper published in Toxicological Sciences. So that was the first step. We developed an AOP, and what we then did, and what is still ongoing, is we actually verified the AOP. So we checked its reliability and robustness. This was done in vitro using 3 in vitro models, as shown on this slide. First of all, human primary hepatocytes, which, as some of you might know, is considered as the gold standard in vitro model in the area of liver-based in vitro modeling. I have to say that we are still doing this, or still have to do actually these experiments with human primary hepatocytes, because as far we have prioritized the use of this human hepatoma Hepar-G cells, which are liver cancer cells, and also these human skin-derived hepatic progenitor cells that have been developed or differentiated in vitro to hepatocyte-like cells. So as a first step, which is called AOP verification, we used a compound of which we know very well that it induces cholestasis, and we carefully selected here a drug, bosentan. You can see its structure on this slide. So, which is a drug that is used for the treatment of pulmonary hypertension and which is very well known to induce cholestasis in human patients. In a set of preliminary tests, we first optimized 3 working concentrations, as you can see here, by means of an MTT test, and we also decided to use 3 exposure regimes, so 1-hour exposure, 24-hour exposure, and that's then also combined with a washout period, so in order to check for the reversibility of the effects. So this AOP verification step actually had 2 main objectives. So the first one, specifically in the context of the objectives of the detective project, was to look for new, or actually for the detection of established biomarkers. So basically we wanted to confirm the occurrence and the existence of the already identified information blocks in the AOP by means of several assays which are listed here on this slide. And then the second objective, which was the most important one, was the characterization of new biomarkers which can give rise to new information blocks for the AOP, so which can be included during further rounds of optimization of the AOP, so in future, in the upcoming time. And actually we were in a very good position to do so because we all had this very nice and fancy omics technologies available in the detective and the SURET consortium, so which really allowed to look for new biomarkers of cholestasis. So that was AOP verification, and then the second major step was AOP application, which is still ongoing. And here, in contrast to the first step, we actually selected a chemical with poorly documented toxicological properties, so of which we actually assume that it might induce cholestasis. And of course, in view of SURET, which was funded by Cosmetics Europe, we selected a compound that is of relevance to cosmetic industry, namely triclosan. You can see its structure here. It's an antimicrobial agent that is used in a lot of consumer products. And it is known in literature, well, it is not known. Actually, we have some hints. We have some indications that it might induce cholestasis, but it's not pretty sure yet. So this is the ideal compound to actually check the applicability of an AOP, in this case of the AOP cholestasis. And here again, we optimized 3 concentrations and 3 exposure regimes. Now, unfortunately, because of time restrictions, I'm not able to present the results of all of this to you, but I can tell you that we are actually now finishing the first draft of the paper. We are very excited about it because in addition to confirming the existing biomarkers and the information blocks in the AOP on cholestasis, we identified several new biomarkers, really unexpected things, but that are really mechanistically anchored and which can provide new perspectives for the identification of new biomarkers for in vitro toxicity screening. So please keep your eyes open in the next few months on PubMed Medline for this paper, which will be submitted, by the way, to Archives of Toxicology. So what's next? So we had AOP development, AOP evaluation and AOP assessment, and then also, as mentioned, AOP verification. Well, what's next? I will illustrate this by means, again, of the AOP on cholestasis, because we have a lot of experience with this. But what will be discussed now, so on this slide, basically is applicable to all AOPs. So what is typically done in the verification round of an AOP is that people usually rely on these omics technologies combined with in vitro models. Now, of course, what you can do is to extend this and also test a number of functional parameters. In the case of the AOP on cholestasis, this could be the inhibition of the BSAP, which is the molecular initiating event. And you can also introduce or also rely on a number of in silico methods, so in addition to the in vitro testing. Now, of course, you can go on in verifying and optimizing AOP, so testing the robustness and the applicability. Now, at some point, of course, you need to decide for yourself whether your AOP is fit for purpose. I will be coming back to this on the next slide. Now, in case of the AOP on cholestasis, the further robustness and applicability testing will be focused on the use of human primary hepatocyte, because as mentioned before, we now have mainly focused on the use of the CPRG cells. And of course, we also intend to elaborate the use or the list of chemicals to be tested. So not only drugs, but also cosmetic ingredients. Now, very important, and I think that the people who are involved in the AOP field would fully agree with me, on the top priority list for the upcoming years is quantification of AOPs. And this can be done in several ways. So for example, by including quantitative structure-activity relationships, or also by considering concentration-response relationships. And this is really, really indispensable if we want to use AOPs for risk assessment purposes. And of course, in the most ideal situation, this can be also, and this has to be complemented with kinetic data and exposure data. Now, as mentioned before, you basically can go on in optimizing and fine-tuning AOP. So an AOP is never finished. It's a very dynamic tool. But at some point, you just need to decide whether or not the AOP of interest is fitting your purpose. So whether or not it is sufficient to actually do what you want to do with an AOP. Now, I think that most of the AOP developers these days think about applications in the field of risk assessment if they develop, when they are actually developing AOP, and this also is the case for the AOP on cholestasis that we have developed. So as mentioned before, one of the most interesting applications of an AOP is to include them in the steer testing strategies or approaches. So there are two things that you can do here. So first of all, AOPs can be used for qualitative testing, yielding a yes or no answer. So basically, what would be done here in this scenario, you would have a chemical of which you suspect that it might induce cholestasis, and you want to run it through the AOP and get an answer on the question whether or not this chemical with, not really a clear-cut toxicological profile might induce cholestasis. So the first tier here could be in silico testing. So just looking to the chemical structure, and just by looking to the structure, you could already tell something about, the potential to induce the MIE, whether or not. The second tier, so next you can move to in vitro testing. And as mentioned before, not only omics, but also functional tests should be considered here. Now, this is exactly why I regret a bit that OECD gave up this distinction between intermediate steps and key events, because this would really, really be nice to apply a kind of weight of evidence approach. So how you should see this? Well, basically you should make a kind of a list of assays that you want to test, so in order to run a chemical of interest through an AOP. So what you can do is just, like ticking a box. So you have to run then the chemical that you want to test through a number of these assays. So if you have a positive result then for an assay that has the molecular initiating event or a key event, well, this should be considered as decisive data, whereas a positive result for an intermediate or an assay that is linked to an intermediate step is rather supporting your decision whether or not this chemical with unclear toxicological profile might or might not induce cholestasis. So that's exactly why it might still be interesting to distinguish between intermediate steps and key events. Now, of course, in a bit farther away future, as already mentioned before, it would be very good, actually that's the ultimate goal, to use AOPs for quantitative testing and ultimately to establish NOAL, so no observed adverse effect level, because this is exactly what risk assessors want to see. But as mentioned before, this is not a standalone thing, so we have to also rely and include on several other tools like physiologically based pharmacokinetic modeling and approaches like quantitative in vitro, in vivo extrapolation. So I more or less came to the end of this presentation, and I just listed on this slide, just for your information, a couple of papers that might be of interest to you for further reading. Well, the first one has been written by Anke. I think some of you, well, most of you are familiar with this. He's considered as the godfather, one of the godfathers, if you will, in the AOP field. And two very nice papers then by Dan Villeneuve, published 2 years ago in Toxicological Sciences that, focus on more the practicalities on how to develop AOPs. And then finally, a paper written by myself that was published 3 years ago, which exemplifies the actual applications of an AOP. So that was about it. I would be happy to answer your questions at the end of this webinar. So thank you very much for listening.
Thank you, Matthew. As we mentioned, we'll hold all the questions to the end, but if you have a question now, please submit it using the question panel on your GoToWebinar toolbar. And we'll move now to our second speaker today, Dr. Barbara Buckley. Barbara joined the National Center for Environmental Assessment at the US EPA as a toxicologist in 2007. She primarily works on the integrated science assessments, which form the scientific foundation for the review of the National Ambient Air Quality Standards. She received a PhD in environmental health science from the Johns Hopkins University, where she trained in inhalation toxicology and subsequently worked in the departments of medicine and pharmacology at Duke University. She has more than 20 years experience studying biochemical and cellular signaling pathways involved in oxidative injury and antioxidant defense in lung epithelium and vascular endothelium. And her research interests include the role of nitric oxide and reactive nitrogen species in cellular adaptive responses. Barbara?
Okay, thank you, Amy, and thank you for this opportunity to speak to everyone about adverse outcome pathways. Our application of the AOP paradigm is a little different than Matthew's, so I'll begin by providing a little background on our work at the EPA. And of course, here's our famous disclaimer. My focus will be on AOPs as applied to the National Ambient Air Quality Standards, or NAAQS. First, I'll talk a little bit about NAAQS, the need for multi-pollutant context, conceptual models, examples from several frameworks, including mode of action, common adverse outcome pathway, toxicity pathways, and then finally an AOP approach illustrated with case reports for assessing criteria or pollutants in a multi-pollutant context. Okay, the group I work with at the EPA is responsible for the integrated science assessments, which we'll call ISAs, of the criteria air pollutants regulated under the Clean Air Act. Development of the ISA, depicted here in the red box, is an early step in the review of the NAAQS for these criteria pollutants. The ISAs serve as the scientific foundation of the NAAQS review process. They provide a comprehensive synthesis and evaluation of the most policy-relevant science from several disciplines. Health effects evidence is provided by epidemiology, controlled human exposure studies, and animal toxicology. And for the criteria pollutants, there is a wealth of available human data, both experimental and observational studies. Some key points are that the NAAQS are promulgated for individual criteria pollutants, and they are particulate matter, ozone, oxides of nitrogen, sulfur oxide, carbon monoxide, and lead. And today you'll hear me talk about SO2, sulfur dioxide, and NO2, nitrogen dioxide. And they are the indicator species for sulfur oxides and oxides of nitrogen. So there are two sets of standards in the NAAQS. There's a primary based on health effects and a secondary based on welfare effects. And one of our criteria pollutants, particulate matter, is actually a mixture. Currently, NOx and SOx are being reviewed together for the secondary NAAQS. And this is because of a conscious shift away from a single pollutant focus towards a multi-pollutant focus. About a dozen years ago, EPA received some advice from the National Academy of Science to review and develop NAAQS for related pollutants in parallel and simultaneously. That led to a lot of discussions and a workshop held in 2011 to consider multi-pollutant approaches to scientific and risk assessment, and it was summarized in this review paper. A major point coming out of this workshop was that progress in evaluating the effects of single pollutants in a multi-pollutant context could be made using existing scientific information and statistical methods. But to advance the field, concrete steps were needed in exposure science, epidemiology and statistics, modeling and risk assessment, and toxicology. For toxicology, it was recommended that pollutants be grouped according to their common modes of action for assessment. At about the same time, a couple of investigators weighed in on grouping of pollutants and their effects. And the grouping was based on common pathways, such as by fundamental biological reactivity or by surrogate marker. So the workshop led to this project, and it asked a simple question, which is important. Do multiple criteria pollutants act through similar pathways to induce health effects? And that led to examination of a couple of different frameworks that could be used, as I'll describe in the next slide. Of course, everyone knows about mode of action, and Matthew has talked about it quite nicely. And it is a sequence of precursor steps, and it involves measurable key events. We applied the mode of action paradigm for the respiratory effects of ozone. This information was taken from the most recent ISA. It begins with inhalation of ozone and its reactions within the extracellular lining fluid in the respiratory tract, which lead to other key events, endpoints, and clinical outcomes. As you can see, the scheme is pretty complex. It's not simple, it's not linear. The cause and effect relationships are indicated by the arrows. The dashed and the solid arrows represent different degrees of certainty about the pathways, taking into consideration the strength of evidence from human and animal studies. And here we have the human depicted by the solid arrows and the animal evidence by the dotted arrows. So as you can see, ozone has multiple modes of action, and they can be tied to different clinical outcomes. Important point. Ozone, like other reactive gases, forms secondary oxidation products in the lung lining fluid. And this is a key step, and it's not likely mimicked by in vitro systems. Okay, so the MOA paradigm can be adapted to incorporate converging pathways or converging effects of multiple pollutants, as you see here. And here we have converging pathways approach for 3 criteria air pollutants: sulfur dioxide, particulate matter, and ozone. The pathways are color-coded, and essentially we're integrating information about 3 criteria pollutants in a key events analysis. And the advantage of this approach is that it's very visual. It enables us to see which pathways we have evidence for in the case of each of the pollutants and where there are common modes of action. Here's another example, and we have some emerging studies providing evidence that the inhalation of particulate matter and ozone lead to cardiovascular effects via 2 main pathways. One is involving pulmonary inflammation leading to systemic inflammation, and we think that may be caused or involving diffusible mediators. And the other is activation of sensory nerves in the respiratory tract leading to altered autonomic function, and we've referred to that as neural pathways. Okay, another framework or paradigm is the common adverse outcome paradigm. And it was developed by the NRC to guide the US EPA in conducting a cumulative risk assessment of a class of chemicals, in this case phthalate esters, which share a common health outcome rather than a common mechanism or mode of action. And it broadened the focus to include contributions to the common health outcome resulting from stressors other than phthalate. Now here's an example, an illustration, showing phthalates and other stressors as the input with altered male reproductive outcomes as the output, and an integration point, disturbed androgen action, linking the stressors and the outcomes. And of course, different mechanisms can contribute to disturbed androgen action, including mechanisms affecting the level of the hormone and the ability of the receptor to respond to the hormone. This sort of thinking can be applied to air pollutants and cardiovascular effects. In this example, the progression of atherosclerosis is the integrating endpoint which links the pollutants to the common adverse outcomes. Where here altered vasomotor function is the integrating effect. And here a more detailed scheme that shows the various effects which are linked to vascular toxicity that have been demonstrated in air pollution studies. So the common adverse outcome paradigm for phthalates was designed for situations where pollutants act on specific molecular targets. But that is not necessarily the case here. But in the case of some air pollutants, altered nitric oxide bioavailability results in decreased flow-mediated dilation. And that is characteristic of endothelial dysfunction, a type of vascular toxicity. And this may result from any of three known pathways depicted here: a decrease in the activity or protein level of the synthetic enzyme for nitric oxide, or increased production of superoxide radicals, which reacts with nitric oxide to form peroxynitrite. So as we see here, the common adverse outcome paradigm may be useful if you have specific molecular targets. So you all know this document. It was published in 2009 and it was based on the NRC report, Toxicity in the 21st Century. And it described the toxicity pathways as cellular response pathways that, when sufficiently perturbed, are expected to result in adverse health effects. And the key elements are a sequence of precursor steps, toxicokinetics and dynamics, perturbation of normal biological pathways, and adaptive responses. However, it has a limitation, and that is there's a very large gap between cellular events and morbidity and mortality, which highlights the need for validating or anchoring upstream events to downstream clinical outcomes. A good example of this toxicity pathways approach is applied to air pollution research. It's found in this paper by Chanel et al. And it combines in vivo exposures of humans with in vitro assays. In this study, human subjects were exposed to 2 kinds of air pollution for 2 hours by inhalation. Blood samples were obtained both immediately and after 24 hours, and plasma samples were prepared. Some of the plasma was analyzed for SLOX, and part was applied to the cultured human endothelial cells. Media from the cultured cells was analyzed for pro-inflammatory protein IL-8 and for the mRNA of endothelial adhesion markers. And the study demonstrated that circulating factors, that is, components in the blood of the volunteers, were altered by exposure to NO2 and diesel exhaust. These circulating factors led to endothelial cell activation as measured by the in vitro assay. This is really interesting work, but more is required to link these changes to morbidity, since we don't understand the relationship between SLOX, interleukin-8, and adhesion molecules, and cardiovascular outcomes. However, these biomarkers reflect pro-inflammatory and pro-oxidant status. In this approach, you can see it can be used to evaluate the effects of different mixtures of criteria air pollutants. In this example, diesel exhaust is a mixture, and one component of that mixture is NO2. And in fact, in the study, it compared the responses to the NO2 alone to the responses to the whole mixture. All right, Matthew's already introduced the AOP pathway paradigm, but I'll say a few words. At one point is that the molecular initiating event could be a ligand-receptor interaction, or it could be a non-specific effect such as reversible hydrophobic interaction or protein oxidation. And that makes it different from the common adverse outcome pathway paradigm. Biomarkers are a key part of the AOP paradigm, and here they're denoted in blue boxes. They can be at the cellular response level or at other levels, and they can be defined broadly to include biomarkers of exposure and biomarkers of effect. And importantly, a biomarker does not need to be directly linked to a downstream event. As you can see here, enzyme induction may not be directly linked to anything downstream. And of course, in this paradigm, we have the certainty of the relationship denoted with different types of arrows. 2 more points about AOP. It can be applied to chemical mixtures, and a limitation is that toxicokinetics is not incorporated. We decided to apply the AOP paradigm to the situation of air pollution mixtures and asked, do multiple criteria pollutants act through similar pathways to induce health effects? This work by my EPA colleague Ayman Faraj and I was published in July in Toxicology. As background, you may be aware air pollution is a complex mixture of particles and gases, and conventional EPI and tox approaches can't evaluate all the possible unique mixtures. A large number of tox studies have been conducted to elucidate underlying changes, but their utility is limited because it's difficult to link the changes to an adverse outcome that is relevant to risk assessment. So we thought that this type of mechanistic data would be most informative when translated to measurable changes, including organ responses, clinical consequences, and impacts to the population at large. So our goals were to develop a conceptual model for air pollution mixtures that links initiating events at the cellular and molecular level to population-wide impacts, and to identify measurable endpoints that could integrate the effects. We chose airway hyperresponsiveness, a key feature of asthma, and we talk about it in case report 1, and endothelial dysfunction, a risk factor for cardiovascular disease, and we discussed that in case report 2. These are both physiological changes at the organ level, which can be measured in the clinic and the laboratory. And we incorporated information from experimental and observational studies. Most of that information came from the ISAs. In the case of case report 1, we considered irritant gases such as ozone, nitrogen dioxide, and sulfur dioxide, airway responsiveness, which reflects the sensitivity of smooth muscle to natural and to natural or pharmacologic stimuli, and respiratory morbidity. And many epidemiologic studies showed associations between pollutant exposure and respiratory effects associated with asthma. Short-term studies were generally linked to asthma exacerbation, and long-term studies were sometimes linked to new onset asthma, or in other words, increased incidence of asthma. We had evidence from the controlled human exposure studies to draw upon, and they generally involve exposures of 1 or 2 hours, with effects most often measured immediately or within a few hours of exposure. But some studies measured effects up to 24 hours later, or repeated exposures over a few days. Generally, the results can be grouped into 5 categories: formation of secondary oxidation products in the lung lining fluid, increased immune responses in healthy individuals and in allergic asthmatics, physiologic changes in airway smooth muscle in both groups, and an increased inherent reactivity of the airway smooth muscle in both groups. And the tox studies sort of confirm and expand that. Most of those studies involved exposures of a few hours, but a few involved exposures over several weeks or months, which of course you can't do in humans. These resulted in morphologic changes such as airway remodeling and disruption of the epithelial mesenchymal unit during lung development. Generally, the tox results could be grouped into similar categories as in the controlled human exposure studies. But one caveat is in rodents we don't have asthmatics, and so we used animal models of allergic airways disease as a phenotypically similar disorder. So we took all the information from the previous 3 slides and integrated in this one, and that included evidence from both the short and long-term studies. There's a lot of overlap in the effects of the 3 irritant gases, all converging on airway hyperresponsiveness, indicated by increased airway smooth muscle contractility and sensitivity to agonists, which is an intermediate endpoint at the organ level, and it's linked to individual level responses such as reversible airflow obstruction and population level responses such as asthma-related hospital admissions and emergency department visits. Most of the evidence was from the short-term studies and points to asthma exacerbation. However, some evidence is from the long-term studies and points towards the induction of asthma. And you'll see that down at the bottom of the response box. We found persistent airway inflammation and remodeling, impaired lung development, and adjuvant effects on allergic sensitization in several studies that could certainly be tied to induction of atopy and development of asthma. And we discussed a second case report, one that dealt with the effects of PM and ozone exposure, endothelial dysfunction, and cardiovascular disease. And we define endothelial dysfunction as impaired blood responses to vasodilators, and it can occur in a variety of blood vessels. So there were many epidemiologic studies which showed associations between pollutant exposure and cardiovascular effects. And some of the short-term studies showed associations with acute events like MI. And some long-term studies showed associations with the progression or development of atherosclerosis. And short-term exposure studies also showed associations with endothelial dysfunction. And they involved exposures of a few hours or up to 1 day of exposure. And there was a lot more evidence from the epidemiologic studies for PM effects as opposed to ozone. From the controlled human exposure and tox studies, we saw endothelial dysfunction as a result of exposure to both agents. And most of the studies involved short-term exposures, but there was a study of long-term exposure to particulate matter in rodents where they developed atherosclerosis, and the atherosclerotic vessels exhibited endothelial dysfunction. So a key question is how inhalation exposure to these criteria pollutants led to systemic effects. And there have been many studies of PM exposure that suggest that pulmonary inflammation and oxidative stress mediates systemic inflammation oxidative stress. And there's a less evidence of this for ozone, although it may also be the case. There was one key study of ozone exposure that I'll describe. It was an in vivo exposure to ozone coupled with ex vivo assays involving isolated blood vessels. It was similar to the study by Chanel that I talked about earlier, except that it was conducted in rats and isolated blood vessel preps were used. And the results showed that circulating mediators contribute to the impaired blood vessel responses, and also that circulating mediators acted by decreasing nitric oxide bioavailability, which we discussed earlier as a mechanism underlying endothelial dysfunction. So the information from the previous slides is integrated in this one, and the molecular initiating events are likely to be oxidant-induced lung injury and inflammation resulting in systemic inflammation and oxidative stress. And the cellular responses we talk about here are cellular responses in the vascular cells. There's a lot of overlap in the effects of particulate matter and ozone, converging on endothelial dysfunction indicated by altered endothelial-dependent vasodilation, which is an intermediate endpoint at the organ level and which is linked to individual-level responses such as MI and population-level responses such as hospital admissions and emergency department visits for cardiovascular disease. Most of the evidence is from short-term studies and points to MI. However, some evidence is from long-term studies and points towards the development of atherosclerosis. So in this model, we have developed may facilitate risk assessment by providing a framework that can be used to converge the effects of air pollutants based on common underlying mechanisms and to identify data gaps to enable prioritization of research and to allow the incorporation of biomarker data. But it has some limitations. For example, the population effects may be mediated by alternative mechanisms than the ones we've identified. It does not account for adaptation and repair. It does not address dose-response considerations, and no toxicokinetic or toxicodynamic data have been incorporated. So a previous speaker in this series gave a talk, What a Difference the Dose Make. Annie Jarbeck made the point that dosimetry links exposure and response. Matthew's made the same point. And we also need to keep in mind that quantitatively describing dose metrics associated with key events is required to advance the AOP field. So in conclusion, our approach facilitates the evaluation of health effects resulting from air pollution mixtures, and we combine evidence from the different disciplines, and we can develop simplified or more detailed AOPs and indicate the certainty of the mechanistic linkages, and incorporate biomarker information. And in the future, perhaps, we'll be able to incorporate toxicodynamic and toxicokinetic information, and that might lead to a quantitation of exposure-response relationships of multi-pollutant mixtures. And all of this is an effort to advance EPA science assessments. Okay, that's all I have, and thank you very much.
Okay, we are a little bit over our time now. Stephen, would you like to ask any of the questions, or would you rather hold them to send a written response?
We actually have one question that's come in and a couple of comments, and so I don't think this will take long for those who can hold on. The question is for Matthew, although I think Barbara touched on this a little bit, and that is the uptake and or excretion of a compound important in the research? For example, if a compound has low bioavailability, the toxic effects of the systemic circulation bloodstream could be low as well. And sort of as a follow-up on that, do you use in silico models and or other models or animals in estimating the bioavailability?
Yeah, well, it's very important, you know, the concentration and the dose that is being used, and that's exactly the kind of issues that we are facing in general with in vitro testing. It's like, you know, the nominal concentration is not really, so actually what you add to a cell culture is not really identical to what the cell actually sees and what is taken up. So yes, this is a very important piece of information. The problem is that there are no like general rules. You know, you have to like determine this on a case-by-case basis, which is very important. But this is exactly what I wanted to stress at the end of my presentation by including kinetic data. So this also includes biokinetic data, and some of this, to come back to the second part of the question, or to answer to the second part of the question, can be modeled by means of a number of in silico techniques. Now, me, myself, I'm not really familiar with this kind of techniques. I do know that they can be used for answering exactly these kind of questions. And of course, this is what we are actually doing right now to further optimize the AOP. Because again, this is all in view of quantification of AOPs, which is really, really important in, if we want to use AOPs for risk assessment.
Okay, thank you. And there's another question that's come in that's more of a logistic one, which I'm sure everyone's interested in, and that is whether the presentations are going to be available to attendees. The entire webinar has been recorded and will be available. Amy, are the presentations available as separate files, or do people need to watch the recording of the webinar?
A PDF of the slides are actually up on the website now at that first link on the screen, and then the recording should be added later today or tomorrow.
Okay, so the slides are available now, and the complete recording will be available within the next day or so. Right. Then there were two comments that didn't actually have questions, but were just comments. One was that triclosan has a well-documented toxicology profile, as well as the AOP that was identified, that we identified, and this is from Tim McMahon, was the effect on thyroid hormone. And so, Matthew, as you were mentioning, there are lots of AOPs out there, and you mentioned some of them, but certainly not all of them that are out there that either have been developed or are in process.
Yeah, of course, I fully agree, but maybe I have to correct myself, or I had to be a little bit more clear. I mean, for triclosan specifically, that it only has suspected cholestatic properties. Of course, it has many more well-documented toxicological effects, but I was specifically referring to cholestasis. And I have to add to this that all of this was actually done like 2-3 years ago. In the meanwhile, we have some papers, or some papers have been published on PubMed, on Medline, which really further point to the fact that triclosan might have cholestatic effects by interacting with these nuclear receptors. So it only further substantiates what we already anticipated, but I fully agree, of course, with the remark that has been made.
Those are all the comments that have come in so far. So, Amy, I'll turn it over back to you.
Okay, thanks, Stephen. I just wanted to thank our speakers again and to everyone for joining today's webinar, and we look forward to meeting again next month where the topic will be three-dimensional lung models. And with that, I'll end today's webinar and enjoy the rest of your day. Bye, everyone.
Bye.